heard a lot about rare variants, not just in autism, but intellectual disability and so on. And obviously, a lot of folks, the number of individuals on the spectrum or with intellectual disability that have a frank, clear mutation is still a small fraction. And it's a real issue, I think, for families who say, that's really great, but 80% of us don't have that, anything there, not even the specific gene you're looking at. So how do you kind of think about the transition, or how do we think about idiopathic autism in a way that's actually going to be as exciting, dynamic, and fruitful as we think these rare disorders are? So just for yucks, Bill, you get to go first. And anybody wants to jump, we'll ask a few of you. That's one of the most important questions I think we have to answer.
Well, thank you, Joe. At the outset, I declared myself the least knowledgeable in autism, so I want to thank you for that. But I will comment on not the specifics, but again, it depends upon how you look at this. I mean, one could say that about arthritis as well, and yet look at the innovations and look at the therapeutics that have come in the field of arthritis long before it was subsegmented into 400 different diseases. I would also argue, the world I live in is the translational drug development space. And I already spoke about the common enemy being lack of money. One of the advantages that you have in idiopathic autism is it's large. And because of that, it attracts more. I found it fascinating when Jordi commented on the search for things that work symptomatically, which is a bit antithetical in this day and age. And it's encouraging to hear because if one can get at those symptoms, that you can have the opportunity to develop something more broad, even without the genetic marker. I will tell you, as a scientist, and I already said, we're always in a room with people with 200 plus IQs. We love understanding that biomolecular dart and being able to throw that specific dart. We love it. And my colleague to my left is showing you exactly how to do that. Scientists love that stuff. And yet there's a big world out there. So I think that approaching it symptomatically still gives one a lot of hope that there's opportunities for further development. And I, for one, am thrilled by what I heard today.
Want to take a crack at it?
Yeah, it's a difficult question. So on the slide, there was a slide that I showed that kind of divided idiopathic autism and syndromic autism and what people are looking in the clinical trials. And what you can see is that currently, probably, in the idiopathic frame, people are looking at things like behavioral or social deficits, and also anxiety, for example. And with medicines that modulate the cannabinoid system or others that modulate oxytocin or vasopressin. And so if you look at that and you think about that other graph that I showed that kind of splits the different symptomatology associated with autism, I think that's probably the way to think about it, is kind of going back to some of the questions that have come up today, is what are the aspects that are more relevant to those individuals? And if it's anxiety or panic attacks, for example, or lack of sleep, I think that's probably the way that we will have to think about it in the idiopathic space. I think also in the syndromic, but in the idiopathic space that becomes much more relevant.
I just want to add to that, there is an interesting phenomenon in pharma industry that occurs once you find a drug that works. And Bill knows this much better than I do, and he's referred to development in arthritis and how much change we have seen there. Once you actually have a molecular entity that produces an effect and has efficacy, discovery becomes much easier because you can measure the changes, you see how different aspects of the disease cluster together, which components get ameliorated by certain treatment. There is a tremendous impetus to research in that area as well. And then there is a much greater level of willingness to take the risk to develop a drug that works further in a larger population, to repurpose, to open up new avenues. So I think that's what's going to happen in autism and in neurodevelopmental disorders in general, as we start to develop new treatments that actually show improvement, we'll better understand the physiology of the disorders, and we'll better understand how to modulate them. So I think that there is a lot to be said about having that initial success.
I want to add one comment about, and this is from the targeted perspective. Idiopathic autism means that we just don't understand its cause. And our understanding of the genome is really limited to the parts that make proteins. There's a huge chunk of the genome that we don't understand well yet. And so I suspect that there will be a better understanding of the rest of that genome as we go forward. And so while I think it's hugely important to be optimistic and to look at symptomatic approaches and repurposing drugs and things like that, I think we also have to think about the long game, and we need to keep looking for the causes of autism in idiopathic autism, because it's not only going to open up the opportunities for that one person, it's actually going to open up opportunities for other people, right? One proof of principle tells you how to do the similar things for a bunch of other types of disorders.
The only thing I would add is just don't get caught in applying, as we talked about, like one outcome measure to so many different types of disease, right? Because not everybody's going to be able to reach that bar. But if we make several different bars and allow people to get to them in their own way, oftentimes that can happen too.
And I think it's good that we heard a lot of voices on this question. It's actually been asked a few times in this deck. I think it's the question that I struggle with almost the most, right? Most of the people who come through the Seaver Center do not have a frank mutation that I can point to. And I don't want to ever feel that we are not serving them as well. But I do think, as you've heard, a success, any drug that works will be tried in a greater group of individuals. This is the way to kind of get everybody excited and moving forward, to bring resources into the game. I do think also that what you heard about biomarkers, is a way of saying, okay, so, genetics is only one way to stratify patients, and it may not even be the best way. It just happens to be what I do, so I like it a lot. But there's going to be neurophysiological biomarkers that are going to say this person, for example, and I think this is what Paige is working on in our center and other groups as well, there seems to be glutamate deficits in this subgroup of individuals, but GABAergic deficits or excess GABAergic activity in this group of individuals. So, the neurobiology is at some point going to add to our ability to stratify and say, okay, in the big pie of intellectual disability, autism, and even epilepsy, we can kind of, the term I'm now using is biotype, right? That there are biotypes in these disorders that will respond very well to a specific drug. And genetics is just the current state of the art for stratification in terms of what we can do really well because of new technology. But neuroscience will do a much better job in the future. So we did mention biomarkers, and there have been a couple of questions about, what progress is there for biomarkers and how are they actually being used in drug trials? And this is not an easy answer, but maybe we start with you. I mean, obviously, I think I know what you're going to say, but take it away.
So actually, Takeda has a specific group designed to identify, develop, and validate biomarkers in the clinic. And the way we take drug development, and we're not talking here specifically about neurodevelopmental disorders, but across the space, is to begin with, and this may sound a little bit counterintuitive, so hold your horses. To begin with, we don't care whether the drug has efficacy or not. I mean, we do, but the most important thing to start drug development in a human is whether we can detect a biomarker and whether we can modulate that with our drug, with our medicine, in the direction that we think the mechanism is indicating. Once we have that signal, then that opens the gate to increase investment, and you've seen this morning, the amount of money that is necessary to do this type of work. That opens the door to subsequent studies with the drug, like proof of concept studies in humans, et cetera, et cetera. But we have a very strong belief that if without a biomarker, that we can look at and that we can modulate, it's very difficult to justify levels of investment of such a level. So that's kind of, biomarker is central for us in terms of drug development, absolutely. And I assume a lot of other companies feel exactly the same.
I mean, I think it's probably especially important for things that are developmental, that are complicated. If you think you're going to measure, if you're expecting a profound change in language over the course of a typical 12-week clinical trial, it's probably not going to happen, but the biomarker may move. And if it moves a little bit, then everybody's going to start investing in that same clinical trial for a year rather than 12 weeks. Any thoughts about biomarkers and where they're going?
I mean, I can speak a little bit from the Angelman Syndrome experience. The Angelman Syndrome Foundation and the NIH has invested in finding biomarkers that translate across animal models to humans. And so we've looked at EEG. There's an EEG signature for individuals with Angelman syndrome, and that same EEG signature is seen in the mouse model. So we're looking at that, about how rescuable that biomarker is, as well as white matter deficits that we can see in both models. And so I think that those are great because they're unbiased. They're objective measures that we can look at. And then, I think there's another type of biomarker. A lot of people are used to looking at things in the blood that would change. And I think of that in terms of like target engagement. And I think, Ionis and Roche has really led the way for Angelman syndrome in this, and that, they're specifically looking at, can we increase UBE3A? And that's straight up the most important biomarker for is the drug doing its job? That's my 2 cents.
Sure. And what about in Duchenne or muscular dystrophy? People trying to find things that work, muscle mass, things that they can use as a very quick outcome and then decide if it's worth the further investment?
Yeah, I mean, there are lots of academics and companies looking at things in the urine that might be changing. We're looking at fat in MRI, for instance, things like that. But, it's important, I know, but, 0.9 dystrophin is a really small amount of dystrophin to approve a drug on, right? But it did indicate some clinical benefit. While it wasn't the clinical outcome measure that was the primary outcome measure. And so I think that, sometimes we're going to see change in the short term too, that can correspond with the biomarker.
Yeah, no, I think that's a. I don't mean to be insulting.
Dangerous area to go.
But it's
I know.
But in some ways, strength and muscular activity is a little bit easier to put together compared to language, cognitive abilities, and so on, in terms of how long you need to see an actual change.
Yeah, and I think it's really interesting. So we've been looking at Angelman's as well for quite a little bit now, and I think that, like you saw in the videos, and I think that videos and home video capture especially. When parents of kids with AS are at home, that they actually may be the best assessor to start to pull those things out in the short term and indicate whether or not there's benefit. And so we know when kids with lots of neuromuscular disorders and lots of other disorders travel to sites, when we have different assessors, when we do things with our kids, when we get to clinic, that's unusual. We can have a change in behavior, we can have a change in speech, we can have a change in cognition. And so what we advocate for in Casimir is really taking a look at that patient in their home environment, creating standard operating procedures, and then looking at language and cognition and behavior, and then having KOLs come in and do like a CGI-C scale to see whether or not it would be probable that they would have those changes in that time, because it just may not be detected in clinic and correspond with your biomarker.
Absolutely.
And you don't want to miss it.
You raised your hand.
I did, and it was in particular because I wanted to get Christine's thoughts on this. This is a rare treat for me, because I know in the room we have parents, we have families, we have people who live with these diseases, and we get into these hyper-technical discussions about biomarkers. I live in a space where if you don't have a biomarker, we're not going forward. And I bet you and I both cringe in horror at that on some level, but I sort of view it as, well, if those are the rules of the game, I can play that game. And if I have a hypothesis that something is really going to work, I will find that biomarker one way or another. And I have to commend you. I love your approach of going after, in a focused way, an outcome that matters and arguing that maybe we don't need. See, the argument against that is, well, you need 40,000 patients. It's just not feasible. And I know you've heard all this stuff. And yet you chose to fight the fight. So good for you.
Because we don't have 40,000 patients, and Angelman syndrome is a really great example of that, where you could give a compound to a family and a mom is going to scream up and down to her. Well, first of all, she goes into the clinician and she's like, she can say her THs. And the clinician goes, okay, that's great. Excuse me, psych coordinator, we don't have any place to document that. There's no, what do I do with it? Do I call the sponsor? What do I do with this? But yet, okay. Or the mother comes in and says, he's never used the potty by himself. He went and used the bathroom by himself. He doesn't, 3 days in a row. And the clinician goes again, well, that's not what I'm supposed to be measuring. So does it not count? And we and especially in rare disease, we can't let ourselves go down that circle, because we'll never get drugs that are really important to families approved and given that time, so that when gene therapy comes through, we've got some kids that are healthier than they would have been, or even alive. And so I think, I think we really got to think about, especially in rare disease, how we approach things with biomarkers. And I'm all in with the biomarker, but I'm far more in, because you still have to prove that biomarker leads to clinical benefit. I'm far more in with seeing functional benefit with patients and families at home, and you can standardize anything at home, and you can quantify almost anything out there, and that's what I've learned in rare disease so far.
I just want to second that. I'm a biomarker person, I spend a lot of time developing biomarkers, working on implementing them in complex clinical trials. But the bottom line is it can become a self-fulfilling prophecy. You need that biomarker to convince the investors, to convince your management that this is something you can measure, and you don't have to embark on either a very complex endpoint such as behavior or language, or on a very long-term outcome that will take 10 years to assess, like survival due to reduced cholesterol or something. But what's important is really to keep that approach of understanding how the biology ties with the function, how that ties with the quality of life. And pretty much anything is quantifiable, as Christine said. You just have to think about ways to best implement that in the particular patient population.
Because I will just add, with the digital revolution and the wearables revolution, many of these biomarkers or measures, or whatever we want to call them, will likely be more objective and more, you can standardize them across patients. So that's something that we definitely need to take advantage of.
So each one of those videos that you saw, at least in Duchenne, and we're working in other rare diseases. So those are all now done under SOPs. Patients, caregivers are capturing those videos at home, and then they are scored by physical therapists who are blinded to the time, blinded to the patient, and we actually can score them and come up with a severity score as a result. And so we've taken that approach into other disease types, and then taken a lot of our qualitative work and brought that down as well into a quantitative now way of quantifying things, measuring things.
So there's a question here about whether ASD or autism qualifies for orphan drug status. So I think the answer to that is probably no, or definitely no. But then the flip question when it goes on to say, so, are there ways, verbal, nonverbal, to stratify autism to make it as amenable to a orphan designation? And so clearly rare disorders are one way, but what are some other ways of taking kind of these complex heterogeneous disorders, schizophrenia, autism, and so on, and stratifying in such a way that there might be a very clear subgroup based on phenotype?
Oh, may I?
Sure.
So orphan drug is actually something I know a little bit about, so it can be dangerous. So the rules are interesting. It basically caps the prevalence at 200,000 individuals in the U.S. There's also a less-known provision that says, or if the company is not going to be able to make this commercially viable. So, you know, there are avenues to argue this. Now, I mentioned during my talk that this has become controversial, in part because the FDA gets embarrassed when people use orphan drug rules, and then the company winds up making scads of money. Lipitor was first approved as an orphan drug. So that's the kind of thing that leaves a bad taste in the agency's mouth. There is more than one path that provides orphan drug exclusivity, although it's tougher to argue the latter point. And like cancer, cancer is not one disease anymore. It's thousands of small diseases. So as we get more and more into precision medicine, it is what your labeled indication says. So if your label says you've got this genetic mutation, and that happens to be a group less than 200,000, you do qualify. So I think that there's more opportunity in the autism space to get those privileges.
Your mentor and mine, Herb Conrad, said everybody is, what was the term you used? Everybody's looking for, they're orphanizing their disorder, right? They're taking schizophrenia and then slicing a bit out of it and saying this is a subset.
The good thing is that the regulators are more open to it. I mean, if you think about cancer that just got mentioned, a couple of years ago we got the first approval based on the genetics of cancer, not the site, not the affected organ, based on microsatellite instability. So I think the agency is becoming much more open to redefining indications to better fit your drug, because the drug was going after this microsatellite instability and restoration of the downstream pathways of it. So the importance is how you define your population and where you position your drug.
I'm going to answer the next one because it's just about my level of ability to answer. Where can we get copies of your slides? So we have videotaped this, and we're going to post everything online. Look at the Seaver website. It takes a little bit of time. It won't take that long. But all the talks will be available online. And I hope you all signed your waivers, now that I think about it. Another question is also, some of these gene mutations, are they associated with specific symptoms? And the answer is probably not, right? So a lot of the things we're talking about in the NDD space, like language, motor, presence or absence, or even autism, epilepsy, they're fairly nonspecific. It doesn't mean that some of these disorders that we're talking about may have more of something or less of something, but you know, nothing we know phenotypically is pathognomonic, if you will, actually that tells you this is blank, or there are very few examples of that. And that's why I kind of, in my own mind, I think of autism, intellectual disability, and even epilepsy, or severe epilepsy, as part of a continuum of disorders that share genetic risk, is as different as they may be clinically. And for me, I think that's almost a virtue, because you know, we're looking at a larger group of people, and anything we do is going to help a larger group of people. I could be wrong. What do you guys think? Who'd like to jump in on that and tell everybody how Joe is wrong?
I mean,
Knock
you know,
yourself out.
I'm lucky because I get to focus, right? And that said, when we focus, we can take two individuals with the exact same mutation that causes Angelman syndrome and find that they're very different from one another. And so I think that that's kind of an important point, right? If you can take two people who should be very similar, you can take siblings, in fact, with the same mutation, and they're going to have they could have very different presentations. And so I really loved Christine's talk about, you know, starting with an individual as their own baseline, because that's going to be very important. And I know on behalf of the Angelman community, we've been talking to the FDA with that in mind, that you know, one person walks and one person doesn't, and you can't use walking as a outcome measure, because it just might not be realistic. And so I think that that's important to keep in mind, even for broader autism.
Yep.
I mean, I'm not going to add anything that we haven't said before, but I'll reiterate that that's kind of the attractiveness of syndromic forms, is that at least you have a place to direct your therapy towards. And then hopefully, because I don't think we know yet, that will take care of some of the manifestations that appear in the patients. How we will get that approved and how we will interact with the regulators, we'll probably need people with the strength that we have seen today But I think that's the beauty or the, the essence of these disorders, that you can start with something that you can that needs to be fixed.
I just want to go back to the orphan drug comment. So while we do, it would be great if you could get down to genetic mutations and get the orphan drug status for different types of autism, but then there's also in the 21st century cures, there's something stipulated for the extrapolation of data. So then you could extrapolate data from specific genetic types of mutations of autism and apply it to additional types. So that exists within 21st century cures.
Yeah, that's great, and I would also comment, if you ask your average prescribing clinician what the exact labeled indication is for any given drug, they usually don't know. So it becomes, the race is to get your first drug approval. And once you get your first drug approval, hearkening back to an earlier comment, drugs tend to get used more widely, and then you find out that it works. And that is off-label drug use, which is sort of a bad word in the pharmaceutical industry. The pharmaceutical folks are not allowed to sell based on that information, but doctors can prescribe as they see fit. So you can also get a small indication. So I view this, forgive me for viewing it as a game, but there's gamesmanship involved in getting drugs approved. And the game, the challenge, is to get that first drug approval, get it out on the market, and from there you build. And very often you find out that the drugs that can get to that point prove to be much more successful than you anticipated. Excuse me.
So maybe I'll just kind of collapse a couple of the questions together, and maybe this will be the last question. But some of the questions have to do with both academia and pharma. Do they share their successes and failures? There's a I go on clinicaltrials.gov. There are completed studies, but there are no results. So I think we're hearing the frustration of the families that are kind of watching and hoping to know about successes and failures or about results, and not always are we terribly forthcoming. Unless it's a huge success, then it's usually on the front page of something. So how can we do better, and just kind of, treat the families with the dignity and respect that they deserve, that if we finish something, they get to hear about it, even if it's negative, or even if it's, it's stopped in the middle because it was really terrible, right?
I'm really going to go here because the worst thing that could possibly ever happen to a family is to sacrifice their time, their child's flesh, their child's blood, okay? The missed birthday parties, the missed holidays, the car rides, the plane rides, okay? Like the worst thing and the most tragic thing that ever could happen to a family is to participate in a clinical trial and to not know what the result is. That's a tragedy, and it should never happen to a single family who puts their child into a study. I, to this day, still don't know why my son has 3 huge welts on his stomach. I deserve to know. He deserves to know. He never wore a pair of pants that buttoned in the front again. And that's not that bad. I have friends who have kids that have had muscle biopsy after muscle biopsy after muscle biopsy, and that data, while the study was put up on a shelf, that data was never released. And that is not fair to do to children. It's not ethical, and it's not moral. And that has to stop.
So I'm loving
Let's have Stormy go ahead.
I'm loving interactions that we've had in our community with the various pharmaceutical companies interested in Angelman syndrome. Ovid sort of led the way in showing how a pharmaceutical company can interact with families and with the patient advocacy groups and the families directly. They're present at all of our family meetings and our scientific symposia. And this example has been followed by other pharmaceutical companies like Ionis and Biogen and Roche. And so we have a huge presence of these pharmaceutical companies, and the families are pressuring them, what's the update? When are we going to see clinical trials? What's going to happen? And so what's kind of cool is I think that it looks to me at least like there's better engagement by the pharmaceutical companies with the families. And so I hope that that translates to making sure that even if there's negative data, it's out there. I know the patient advocacy groups on our behalf, we've encouraged people to publish their negative data. There's a couple of negative data studies out for Angelman Syndrome, and that's really helpful. And then we try to reiterate and communicate with families that those studies were negative.
Yeah. And I think this also comes back to the point that the family foundations, the family advocacy groups have a lot of power, and they should be making this very, very clear that it's not acceptable to them to be involved in studies where there's nothing coming back to them, where it just becomes a black hole. And I suspect in the current climate, especially with the change in regulation and the way that the FDA is listening to the patient voice, those companies that are not giving information back, or those academics who are not getting information back, are going to have a very hard time threading the FDA needle. So just one question,
Yes.
2 questions from the audience, and then we're going to wrap
I
up.
want to thank great presentation. Take it back to 1966 when my son was 3 and he was diagnosed as autistic, severely disabled. Today, at that time, it was 1 in 10,000. Oh, I'm sorry. It was 1 in 10,000. I mean, excuse me, 1 in 10,000. Now it's 1 in 69. Where were all the autistic individuals back in the 40s and 50s when I was growing up? There has to be an outside cause, environmental to a certain extent.
I don't know if I agree, and I know it's a controversial point, but
No, I know. I'm not trying to bring controversy.
no, I'm not. You're not. Yeah, there's no blame there. It's a controversial point. Not you and I. We're not having the controversy. But you know, in 2011, you know, the same methodology that they used a long time ago in the 70s to kind of show that autism was genetic for the first time. In the UK, they did a kind of epidemiological study where they looked for the 60-year-olds, 50-year-olds, 40-year-olds, 30-year-olds, 20-year-olds, and the kids, the younger kids. And they used modern criteria for diagnosis, right? So in the old days, autism was a very, very narrow door. Now it's a very, very broad door. And they asked, what is the rate of autism in the 60-year-olds in England today, who we all know never got a diagnosis when they were 10? And the rate was about 1%. And it was true for the 50-year-olds and the 40-year-olds and 30-year-olds and 20-year-olds. There's been 2 studies like that. You can look at national registries, you see the same thing. Obviously, before somebody coined the term autism, there were 0 diagnoses, right? So the diagnostic criteria, how well you find people, how broad the diagnosis is, and how young you make the diagnosis change your apparent prevalence, right? If 20 years ago you had to be 10 years old to get a diagnosis, and now we can diagnose 3-year-olds, that parent prevalence goes up. You're diagnosing more people per unit population. So I don't claim in any way to be the last word, but there's very good data that would say that there really isn't a huge increase, except, you know, there was a great series of articles, 4 articles about 5 years ago in the LA Times, and they said that it is an epidemic of diagnosis rather than an epidemic of a new entity. So, not to disagree, it's an open question. I think it's a critical one because, of course, if it's all environmental, we should be focusing there. If it's more genetic, we should be thinking about there. You know, that's something that I think we all ponder quite heavily. We don't want to get it wrong and spend all of our time looking for biological causes when it turns out that it's something in the water, right? Which I don't think it is. Good news. If it was. All right, last question.
My daughter, who's now 23, has been on medication since she was 6, obviously starting with risperidone. And she's been on cocktails. And my question is that if there were biomarkers for those meds when they were first created, is there something like a reverse engineering of figuring out why are they working on some people and not others? And the fact that she has to constantly go through these trial and errors to determine, you know, if this is effective. And she's not the best reporter of what's happening. So what about all these drugs that are already out there and, you know, constantly being prescribed for the whole range of symptoms?
So I think the short answer is yes, it has been done, can be done, but it requires to have a biobank. And I know they've got one at the Seaver Center, so you have to have stored samples so that you know where to look and you can follow over time. There's a technical problem, and that is sometimes these biomarkers are very unstable. And unless a sample is stored properly and assayed in the right period of time, you're going to get misleading results. So sometimes it can work, sometimes it won't. I hate to come back to a pet peeve of mine, but I almost pondered the question of, gee, and there are a lot of drugs that do work and they never had a biomarker, so why are we so married to biomarkers now? But the short answer to your question is it can be done, and it has been done, but it's hard.
You can, so then with having that information, you don't have to go through trial and error. You look at the biomarkers and say, oh, that one's going to work for her, but it won't work for him?
It depends on the case. Now it's getting complicated, and I think eventually what we all hope to do is get to a point where we've got enough insight in terms of the analytic markers to know which biomarker is correlated to which outcome that matters, and find out which drug is maneuvering that biomarker in the direction we want to go. So there's a lot of linkages there, but it's sort of the holy grail of what we try to do.
So I want to end on that note, and I want to thank the speakers for absolutely wonderful talks. I want to thank the Tech Demo Group for really exciting things.